forward. So this is section 7.1. So for self-separating theory, so these are our 4 essential questions. We want to look at how has this technology advancement really adjusted what we can learn about cells. We want to look at the difference between microscopes. We want to look at what cell theory is. And the key ones here, the main, if you're walking away with anything, is what is the difference between a prokaryotic and a eukaryotic cell? That's the main #1 thing we want today, because we're going to be looking at exploring at both of these in the future. And then, of course, we have, here's our vocabulary: cell theory, plasma membrane, organelles, eukaryotic cells, nucleus, and prokaryotic cells. So here's our history here. And you don't need to write any of that down, but we know that a cell is a basic structural and its function unit of all living organisms. If you are alive, you have cells. If you remember those 8 principles of life, one of them is cell organization, right? So those cells are so small that we didn't know they existed until we saw a microscope. Just like we didn't know planets existed until we had ways of seeing them as well. Eventually we could use math to figure that out, but same thing, we only thought there was a couple planets in the solar system. Our telescopes can only see so far. You can't see Pluto very well unless you have a really powerful telescope. So they were so small we didn't know they existed. So here's what cell theory is. So cell theory is one of the big things with modern biology, and we have 3 principles with this. The first is that if you are alive, you have at least one kind of cell in you. So all living organisms, doesn't matter how big or small, an elephant, a germ, us, a duck, they're all composed of 1 or more cells. And this is one you will want to write down if you haven't yet. You might have written this down through your notes, and in that case, that's fine. But so all living cells are organized of 1 or more cells. Cells are the basic unit of structure and organization of all living organisms. Again, we have not found anything that does not have cells and does not have cell organization.
What is steel made of?
Sorry?
Cells
sorry, what was it?
like steel, steel.
Oh, steel is no, steel's not cells because it's not living. It's that's like made out of minerals.
Like a wrench.
Yeah,
are those still cells? Or?
No, those ones aren't cells. Those are just like minerals. Yeah.
Like if you look at them through microscope, like what do you fucking what's this iron?
If you zoomed in close,
obviously, but
yeah. Like, you'd eventually see like it would just kind of look like really like it's like sand, like microscopic sand, if that makes sense. Like you zoom in on a piece of sand, like it would look like rough and stuff.
Yeah.
So yeah, we got 3 things here. So all living cells, organisms are composed of 1 or more cells. Cells are the basic unit of structure and organization, and cells only arise from previously existing cells. So cells pass the copies of their genetic material. We have a fancy word for that. Anyone know what it is? DNA. DNA is the acronym for it. I'm not going to make you guys like know what DNA stands for. But so cells pass on their DNA to their daughter cells. That's how they reproduce. Everyone done? Oh, sorry?
Genetic material is DNA.
Yeah. Yeah, and at some point we're going to extract our own DNA. We'll do that in an experiment. So next we look at 2 different types of telescopes. There's more, but these are the main 2 we really need to focus on. The first one's a compound light one. So basically, what's the difference between these? A compound light one uses glass lenses and visible light. So you shine a flashlight underneath it. You have these are those stereotypical microscopes that you think of, like where you can rotate the 3 different types of lenses. Yeah, I'll just load up an image here. So when most people think telescope or microscope, they think this, right? This is an example of a compound light microscope right here. You have those three different lenses, you have a light underneath it, you put a slide in a specimen in, and then it lights it up. So you can usually go up to 1,000 times. After that, it doesn't work as well. Now, most cells, if you look at an actual cell, it's kind of transparent. You can't see it. So what do we usually do is we actually usually will dye stuff. There's some exceptions. You think about a red blood cell, well, it's a red blood cell. It's actually a little bit easier to see. The blood itself is the dye. But if you're trying to find like white blood cells or other things, it's not as easy. So we usually stain them with dyes. And again, you can see an example there on page 180 of your text book. Let me just put that up. Oh, did it not? Oh, yeah, if you look at page 180 of your textbook, you look at the human hand there, you can see how zooming in really affects what it's going to pop up. Let me actually just go into that really quickly. That page 180. And if I look in here, I'm just going to zoom in a little bit, a lot. So again, the more and more we zoom in on this spot here, you can see the skin, then you can see the skin cells. If you zoom in even more, this is just going up by 10 times every time, right? So this is what it looks like to our naked eye. Boom, 10 times bigger, 100 times bigger, 1,000 times zoomed in. These are your actual human skin cells. And then we're going to eventually zoom in a level even further into this and look inside that cell. Where electron microscopes, however, are different. So what we do is we illuminate a sample, we light it up with a beam of electrons, which if you know about electrons are tiny, like one of the smallest things that exist. So we light it up with a beam of electrons, and then those bounce back and we get a magnification. So we can see 500,000 times bigger. You can't do that with a regular microscope. The lenses aren't powerful enough. You don't get enough light. So those electron ones let us zoom in really far. So if you think about like we just looked 1,000 times, zoomed in 1,000%, sorry, 1,000 times into your hand, you were able to see the individual cells, right? Imagine if you go 500,000 times. That's 500 times stronger. Okay. Where's my stuff? Here we go. So we're actually going to look at the difference between a prokaryotic and a eukaryotic here right after. Oh, here we go. I'm looking for my file. Sorry, guys. Found it. Okay. So these are the two types of cells. So prokaryotic and eukaryotic. What do we first notice? Like this one, the one over here is P, prokaryotic. The other one on the right is eukaryotic. Like just looking at it, like what are some of the differences we can kind of distinguish between these?
There's more to the periodic cell.
There's more stuff in it. Yeah. The prokaryotic cell is just it's empty inside. There's nothing there. Whereas the eukaryotic cell looks like it has stuff in it. So we're gonna look at like what actually truly makes up the difference between these two things. So on the left, we have a prokaryotic cell here again, just empty. You see these like little tendrils here. I know this is really zoomed in, so it's not great quality, but you know, there's little like tendrils and stuff here as well. And then this is a eukaryotic cell. So did anyone catch what type of cell this was when you looked in your book?
That was on page 185.
So this one, it looks like it's a plant cell. You can see all the green in here. It looks like a plant cell. So but one thing they do have in the same here, I know it's hard to see, they both have a plasma membrane. So both of these have a plasma membrane around them. So we know there's at least one common thing, but otherwise we just see a lot of differences so far. So let's jump into those. So all cells have a plasma membrane. They have to. That's the commonality between all cells, is they have a plasma membrane. So what's a plasma membrane? It helps controls what enters and leaves the cell. Again, I use the example. Have you guys ever washed dishes too long, took a bath too long, your hands in the water, you pull it up, and what is it? It's all shrunk. So what happened there was your body put water into the environment you were in. If it's in a sink or a bath, it exited from your hand, it went into the sink or the bathtub. It exited through the plasma membrane of your cells. So it's able to pick and choose what it wants to enter and what it wants to leave the cell. If you eat something, you are going to have waste. For humans and mammals, we eat something, we poop it out. Cells have a similar function. Who here took Health Science 20? You did. I know some people online did. When your muscles use energy, it spits out waste as well. Carbon dioxide, lactic acid. Those are things that your body uses up as waste as well. So it needs to choose. It wants to, maybe the waste wants to leave the cell, right? But also maybe it wants to get food. If it's trying to get food, or maybe it's trying to get moisture, you want that to be able to enter. So the plasma membrane is really important because it lets the cell choose what it wants to enter and what it wants to exit. As well, most cells are going to contain some type of genetic material. That's a fancy word for DNA, right? So most cells break down molecules to generate energy. We'll look at more into that later. And then, of course, we have the two types of cells that we just went over, right? We have eukaryotic and prokaryotic. And this is just how you say it. This is just broken it down into its phonetics. So eukaryotic, prokaryotic, and you just combine it all. Okay. So yeah, the big one here is that we know that each cell has a plasma membrane. We know what it does. We know it has genetic material, and we know that they break down molecules to generate energy. So, this. Okay, so let's go into these two types of cells. So eukaryotic cells, the big thing is they have a actual nucleus. They have something in there that is a nucleus, and we're going to learn what those do. But they have a nucleus and other organelles. Organelles are just, organs. It's cell organs. You probably got that from the name. So an organelle of a plant cell, for example, one of those might be responsible for photosynthesis to get energy. So organelles are organs. The nucleus contains the genetic material. One of the big features is that eukaryotic cells are usually larger than prokaryotic cells, usually 10 to 100 times. So they not only are they bigger, they are massively bigger. You can actually take a plant cell, you can look at it with a magnifying glass. You can't do that with your skin cells, for example. You need an actual microscope for it. Okay, so eukaryotic cells are bigger and they have a nucleus and organs. So bigger, nucleus, organs. Three things there. So I'll just make sure you guys are able to finish writing up. We may not get to use the LM or the oops. Yeah, so next we're going into a prokaryotic cell. So prokaryotic cells, those are those simple ones, right? They look a lot more simpler. They do not have a nucleus. They do not have organs. No nucleus, no organ. They're also smaller and they're simpler. So the big thing here is like, these are really simple cells. They're really simple. Like we can tell that just by looking at the two differences between them, right? So they don't have a nucleus, they don't have organs, and they're smaller. So they're probably one of the first things to really exist. Where's my, here, I want this. Oh no, that's a different section that would do that. Okay. Okay. Let's go right in. We're almost done with the notes. Okay, so those are our two cells. So we're going into, oh, I don't think we need to know this. Nope, we don't need to know this. This is fine. We're fine here. Okay, so we're going into these four things here, and now you might not need to write these down. They are in your notes already. So just kind of starting to go through it. Where's my, here we go. I have too many files open. So with this first one here is, if actually I'll give you a minute to write these down if you want. You only need about you only need about 3 lines between each one. Again, if you're feeling confident, that's okay as well. The big one here that's really important is these prokaryotics versus eukaryotics. That's where a lot of what we're going to be looking at in the future is Dell's from.
What are the differences between the prokaryotic and
Oh, so that's what we're going to go over in like 5 minutes. So these types of things we do at the end, they serve as a review, but also these are types of questions that you are asked on your exams and quizzes and finals and stuff. So it's meant to both prepare you for it, but also it's a nice way to review the chapters and sections. Okay, so jumping into this, so where's the mouse? So our advances in microscope technology. So again, without using your notes, try and use your memory here. How have microscopes been useful? What have they allowed scientists to do?
About how our body works.
Learn about how our body works and all living things for sure, but all of science is rooted in what we can detect. And so we needed to be able to see it. So once we've done that, so once we can see them, we can learn more about them. So we've learned more about cells, their structures, and their functions. But basically, like, as a really simple answer is, it's let us see them. If you really want to boil it down to one word, we can view them now and we can learn from them. We didn't know they existed until we got a microscope. Anyone still writing? Not seen anything. So what's different between these two microscopes? We have light microscopes and electron microscopes.
Compound uses glass lenses.
Compound uses glass lenses, and an electron doesn't. Electron uses electrons. And then how else? There's one more big thing. It's in the name. So a light microscope uses glass lenses and visible light. You have to have like a flashlight underneath it or above the specimen to be able to see it. So usually you'll have a light directly underneath it. If I'm looking at skin cells, there'll be a light right underneath it. An electron microscope uses the electrons and magnets to operate and see. So next question here is, we have our principles of cell theory, and there are 3 of them. Anyone remember? Again, try not to use your notes. Try and use your head here.
When your DNA passes on to your genes and
That's one of them. Yeah, yeah. You need to have so cells arrive only from living cells. That's one. Anyone name another one? If you can't, that's fine. That's the whole point of this. All of it has to do with life and living things. All living things are made up of cells. And then finally, cells are the basic structure of all life. So cell theory is basically our theory of life, in that you can't have a living thing without cells. And then finally, last one, what's our difference between a prokaryotic and a eukaryotic cell? So there's primarily three of them. So one size, can you remember which one of them is bigger? Yes, so eukaryotic cells are much bigger, up to 100 times bit larger than prokaryotic cells. Which one of these two is more complex? Prokaryotic or eukaryotic? Eukaryotics, yes, they are more complex. Does anyone know like what makes them more complex? They have things inside of them, right?
Organelles.
The organelles.
Nucleus.
Yeah, so eukaryotics have organelles. And you're right, eukaryotics have a nucleus. Eukaryotics have a nucleus, prokaryotics don't. Eukaryotics are bigger, prokaryotics are smaller, and eukaryotics have those organelles, they have those structures. Prokaryotic cells do not. Okay, so if you guys have that done, then we're good to go. I lied to you, sorry, it was the next section where we're gonna do something on the textbook there. I had my notes wrong.